Okay. All right, so we left off on Tuesday talking about tumor suppressor cells or tumor suppressor genes.
Obviously.
Right?
Naturally.
2 P's and 2 S's. And so tumor suppressor genes, they code for proteins that do what? They inhibit? Good. Yes. They inhibit cyclin-dependent kinases.
Oh, yeah.
Okay, remember we talked about cyclin-dependent kinases plus cyclin, these combos that the checkpoints look for, because the checkpoints check for errors, and then they also look to make sure you have the machinery necessary to carry out the next phase. Well, the machinery necessary to carry out the next phase are these cyclin-dependent kinases plus some particular cyclin, like CDK4 plus cyclin D to go past G1 checkpoint. So tumor suppressor genes code for proteins that inhibit these. Okay, and that's why we call them tumor suppressor genes, because they're inhibiting the machinery necessary to lead to cell division. Trinity, did you have a question? You didn't? Somebody said they had a question, no?
I said
Oh, you did. It was you. Okay. So then the other one.
Your mom. My mom.
Proto-oncogenes. Proto-oncogenes. Proto means first, and then oncogenes are genes related to the cell cycle and uncontrolled cell growth.
That's not one word.
Sometimes. It depends on.
It just looks weird.
I know, it depends. Sometimes it's 2 words, sometimes it's 1. It looks weird when it's 1 word too, because then you have an OO.
Does that say
Like zoology. It's not zoology.
Zoology.
It's zoology. No, it's zoology.
Zoology.
Because if it was zoology, it'd have to have 3 O's.
Rachel, you can vote for zoology.
Wait, what do you call it?
Zoology. Okay, code for
I like it.
cyclins.
Oh, you're right. You're right.
Or, I'm always right. Or other machines that move the cell through
What a workout.
Oh, you don't have anywhere to extend.
Oh, yeah.
its
you
cycle.
have extensions. But
Okay.
you ran the wall.
So proto-oncogenes, they code for cyclin or other machines that move the cell through its cycle.
Can you say it again?
Proto-oncogenes. And again, sometimes it's 1 word, sometimes it's 2 words. Proto-oncogenes. And so both of these are important in regulating the cell cycle. Tumor suppressor genes keep the cell cycle from going when it's not supposed to, right? They keep the cell locked out of the cell cycle in G0, not even in G1, in G0. And then proto-oncogenes code for the machinery that these checkpoints are looking for. Okay? So both of these are oftentimes the targets of cancer-causing mutations. Okay? If you get a mutation in a tumor suppressor gene, you may not inhibit the cell cycle properly. And so it goes when it's not supposed to. Okay? Or if you get a mutation here, you might divide way too rapidly. Even if you're supposed to be dividing, not that quickly. Not that quickly. All right? Any questions here? Because this is it. This is the end of chapter 12. That's literally all she wrote on chapter 12.
Did you hear that?
Well, the current authors of the Campbell textbook
Or a female.
are female, I think, is the lead author is female. But it's Campbell's biology, but no longer written by Campbell, because I think Campbell is no longer with us. I think so. Yeah. All right. Questions? Oh yeah, Michelle. Sorry.
So what's CDK?
Cyclin-dependent kinase. We had that written down on Tuesday. You should see it somewhere written down. Cyclin-dependent kinase. All right. Nothing?
Yeah!
Yeah. All right.
Chapter 12.
Yeah, that's a wrap.
Okay. we are now transitioning over into chapter 13, which I believe is called sexual reproduction and meiosis, or meiosis and sexual reproduction, because I know you like to know.
Emma.
Okay,
Dress like a sweater.
so chapter 13 is called something along the lines, Trinity, of meiosis and sexual reproduction. Yeah.
Wait, that looks like my toes.
It does look like mitosis because it's similar to mitosis. Okay? And so we need to address a couple of questions. Why do we need to have the DNA?
It's chapter 13, right?
Half, like put it in half.
Yeah, isn't that to mean to cut in half?
Yes.
I've never heard that as a verb, to have. It's interesting.
Okay, so why do we need to have the DNA?
I'm done with it.
Okay. I'm pretty sure that's legit. Yeah, to cut the DNA in half.
I like
Why
it.
do we need to have? It's just have, right?
No, half. Like cut it in half.
Oh.
Not like, why do you need to have
To have. You still pronounce it have. To have. Divide into two parts of equal or roughly equal size.
How?
Okay. Have. You still say it's still pronounced have. Have the DNA. Okay. So why do we need to have the DNA?
English language. Because
There's only one process that requires us to have the DNA.
it's reproduced.
Well, only if you're going to reproduce a particular way. Only if you are going to have sexual reproduction.
We don't just like split.
Reproduction. Now, most organisms, by number of species, if we're talking about outside of animals, they do not take part in sexual reproduction. And even many animals do not either. When they reproduce, they make exact copies of themselves. They just go through mitosis, generate some new cells that will go and bud off and form some new individual toys.
To have two DNA.
To have, the
have
DNA. Thank you. Why do we need to have the DNA?
This is definitely a random question.
Okay?
So why when you like sleep the wrong way do you like get like crooks in your neck and stuff?
Because you're forcing your muscles to work in ways they're not supposed to function, because they're like trying to keep your body in proper position while you have it in an awkward position.
Well, how do you like you wake up and everything? Not when you're sleeping it becomes good. Like
Yeah, why doesn't your body like fix it?
yeah, why doesn't let you know like that you should sleep this way? So
Most of the time it does, unless you're laying in such a way that gravity is overcoming your body's tendency to correct itself. And then so your body's trying to correct itself, but gravity's
It's really
holding
getting a
you.
little workout.
He's like going like this, like no.
Okay, so if an organism is going to take part in sexual reproduction, in which two individuals combine genetic information to make an offspring, you have to have the DNA. Because otherwise, you'd end up getting too much DNA, right? So, why do we need to have the DNA? For sexual reproduction, we need to form gametes. We need to form gametes.
again what the lines are.
What gametes are? I'm gonna do this. Sperm and ova.
This is like an anatomy class.
So male gametes are sperm, female gametes are ova. At least male gametes are sperm. In many animals, they're not always sperm. Sperm have to be mobile. In order to qualify as a sperm, it needs to have a flagellum and be mobile. Some of them are just like spermatids or spermatophores. Okay? But we need to form gametes, because the key here is gametes fuse, and this process is called fertilization. That's a Z. Fertilization. So the gametes fuse to form what we call the zygote. the zygote, and this is the embryo.
Fertilizing. That's something's off there.
Oh, today's not going well.
Still there, yeah, sorry. Okay.
Fertilization to form the zygote or the embryo. Okay? And so, how many copies of the genome do you have in a typical cell in your body?
One. That'd be
Well,
Two.
Yeah.
One from your mom and one from your dad.
Yeah.
Okay? So in a typical cell in your body, you have 2 copies of the genome. Well, again, 1 copy that came from your mom and 1 copy that came from your dad. So if you're going to make gametes, you have to take your 2 copies and whittle it down to a single copy. Okay? So that when one of your gametes fuses with the gametes from someone else, it forms a zygote that has 2 copies of the genome. 1 from mom and 1 from dad. Okay.
Are the zygote and the embryo the same thing, just 2 different names?
So the zygote is only at the point where it is a single cell. It is an embryo all the way up until the point at which it becomes a fetus. So an embryo is a more general term. It refers to the single cell state when it's a zygote, and all the way up until it varies from organism to organism where it transitions from embryo to fetus. Okay? So does this make sense?
Yeah.
Okay, so now why do we need to have the DNA? If you're gonna take part in sexual reproduction, you need to form cells that only have 1 copy of your genome. And so you're like, well, which copy are they gonna get? Are they gonna get the one I got from my mom or the one I got from my dad? And the answer to this question is yes. They're going to get some of the material from your mom and some of the material from your dad and some material that never existed before because it's a combination of the 2.
Wow.
And all of that is carried out by the next question that we'll answer. Is this okay here?
Is that
Yeah.
how people get red hair? Hi, Katie.
So red hair is from the production of a different type of pigment. So the typical pigment is called eumelanin, and it's the brown pigment, and then in really high concentrations it can be black. And then there's another pigment, and I forget what it's called, that produces the red coloration. Tori or Issa, which one of you had your hand
I
up?
had
Tori.
a question, but I answered it myself.
Okay. So the next question we need to answer is this one. How do we have the DNA? And the answer to this question is actually written on the board already. Meiosis. Meiosis. And when you see meiosis, you see something that's pretty similar to mitosis.
You
Okay.
can say have and then I write have.
H-A-V-E.
Yeah.
Sorry. That's the English language. It's beautiful. What do you call words that are pronounced the same but aren't the same word and have different homophones? Yeah, pronounced the same but have different meanings.
Those are words that.
Homonyms are they spelled the same but they're pronounced different, like read and read.
I didn't
Oh,
really
yeah.
blue or blue.
Okay, so how do we have the DNA? And the answer to this is through meiosis. Okay? And meiosis, I'm going to show you this process, but basically meiosis, this is meiosis 1.0, because we love this, right? When we do 1.0 and then 2.0. So here's meiosis 1.0. Here's a cell. Man, my circles are getting pretty good.
They really are.
Man, Mr. Freeman only has black expo markers.
He's boring.
All right.
When we get, we just go back down and steal the ones back from our room.
Mr. Lee has like 20 million different colors.
Okay. So here's the cell. Here's the cell at the beginning of meiosis, and the cell at the beginning of meiosis is either I'm trying to remember the name of this. Anyways, we'll call this a germ cell. Okay, so it is a cell that's going to give rise to gametes. There's actually whether it's in the female production of ova or if it's in the male, it actually develops a little bit and has a specific name, but I can't think of those right now. All right, so we've got a germ cell. This germ cell goes through meiosis 1. So unlike mitosis, meiosis actually has 2 cycles. Okay? So in meiosis 1, you generate 2 cells that each only get one of these homologous chromosomes. So let's say this right here is chromosome 1. So we have how many chromosomes again? 23 pairs, 46 total. Okay? So we name them chromosomes 1 through 22, and then the sex chromosomes. Okay, so you have chromosome 1 all the way up to chromosome 22, and then you have the sex chromosomes, which are X and Y. Trinity.
Why are both of those? What do you know?
So these are your 2 copies of chromosome 1.
Why are they both? Oh, okay. Why is there 2?
Because one came from your mom and one came from your dad. Now they were duplicated because you still have that normal cell cycle, right? Where we've got interphase, so you have G1, you have S phase. What happens during S phase? DNA gets duplicated, the centrosomes are duplicated, and so this is when we see chromosomes like this, you know that these are sister chromatids and that they're genetically identical, right? Right? Okay, because you've seen this before. It's just now we're referencing both copies of chromosome 1. Okay, and so after meiosis 1, one of those copies of chromosome 1 is over here, the other one is over there. Okay, after meiosis 1.
So they've divided.
So
They've divided.
separate them?
What's that?
Just separate them?
Well, so now meiosis 2 is basically going to be identical to mitosis. So this right here is meiosis 2, and meiosis 2 is virtually identical to mitosis, but we started with half the amount of DNA. Okay? Because at mitosis, you have both copies of chromosome 1 are in the same cell. After meiosis 1, only one copy of chromosome 1 is in that cell, right? You see that? Mitosis starts here, but meiosis 2 starts here. Other than that, they're identical. Okay? So I want you to remember that. Write that down. Meiosis 2, identical to mitosis. Except for we're just starting with half as much DNA. Alright. I don't have room to draw these cells over here, so I'm going to write continued and with a dot, right?
Why don't we identical to mitosis or meiosis?
Meiosis 2 is identical to mitosis.
Why don't we just call it mitosis then?
Because
It's part of
it's part of meiosis. And here is the continued, and now we've only got one of those chromatids. So during mitosis, we separate those sister chromatids, right, in anaphase. During anaphase 2, which is anaphase of meiosis 2, you separate those chromatids. These chromatids are separated into separate cells. Okay, so mitosis took one cell, produced 2 identical cells, yes? Right? That's what mitosis does. Takes one cell, produces 2 identical cells. What does meiosis do? Takes one cell and splits it into 4 genetically distinct cells. And you're like, wait, but these 2 are going to be the same, right? Because these sister chromatids were identical, right? And then you come over here and you're like, no, these 2 will be the same. These 2 will be different from these 2, but these 2 are the same. Yes, with regards to this chromosome, assuming something doesn't happen, which always happens. But with regards to this chromosome, yes, these 2 would be the same. But when you're looking at all of the chromosomes, because there's some randomness about how they line up. Okay, so this is meiosis 1.0. Any questions?
Wait, so this is like sexual reproduction, right?
This is meiosis to make gametes that facilitate sexual reproduction.
So if you're making 4 of them, all 4 are going to the...
Well, so that's where there's a difference, and we'll talk more about this in meiosis 2.0. But in males, yes, you take one germ cell and you produce 4 sperm. Well, you produce 4 spermatids that have to mature into sperm. But in females, you actually only produce 1 ovum, and then you produce 3 what are called polar bodies. And because the ovum is huge, the ovum is 1/10 of a millimeter in diameter. So it's big enough you can almost see it without a microscope. So if you took 10 of them, yeah, because you can see a millimeter, right? The little marks on a ruler. If you took 10 ova and you put them side by side, they would be a millimeter in diameter.
What are ova?
Ova are the female gametes.
Oh, okay.
Yeah, are the female gametes. An egg or an ovum. Okay.
Kind of big then.
They are huge because when this is happening, the cytoplasm is distributed unevenly. One cell gets like 99% of the cytoplasm when they divide, only half the DNA, but 99% of the cytoplasm of the cell. So, why? Did somebody say why? Oh, because the ovum has to provide all the nourishment for the zygote until it implants, and then once it implants, then at least in a mammal or a placental mammal, once it implants, it can be fed by the mother. Yeah. Emma.
So a male spermatids don't mature, will they not be able to children?
Correct. Yeah, if their spermatids don't mature properly, absolutely. Yeah, they would have a low sperm count and would be sterile. Yeah. Okay. Any questions on meiosis 1.0?
No.
Okay, because it's going to disappear, and then we're going to do meiosis 2.0.
Back to back.
Back to back. Usually it was like one day we did the 1.0, and then the next day we did the 2.0. Okay. So, meiosis. And here we're going to do meiosis 1. So, we're just focusing on meiosis 1 right now in our meiosis 2.0.
Oh.
Yeah, because we're doing 2.0. So we already talked about it, but now we have to do 2.0.
Yeah.
We have prophase 1.
Wait,
Yeah.
Hold up. Go a little faster.
You're like prophase, we saw that in mitosis.
Well, what is meiosis? Wait, we already did meiosis 1, didn't we?
Yeah, we did meiosis 1 in our discussion of meiosis 1.0. Now we're doing meiosis 1 in our discussion of meiosis 2.0.
It's a lot of
So
numbers.
this is now meiosis 2.0.
Yes.
We're focusing on the first part of meiosis.
Look at this. The same phases from mitosis appear in meiosis. Yeah, Trinity.
Oh, so these are the meiosis 1 phases?
Yes.
No, these are meiosis 2.0 phases.
Yes, it's meiosis 2.0, but it's meiosis 1. We're just talking in more
Meiosis 1, we're
detail
just
about
talking
meiosis 1. Relax.
talking about it more, like how do I draw it in my notes, like do I like category?
You just do like meiosis 1 and then underline it and then write out the.
Or do meiosis 1, put a little symbol next to it, like maybe a cross, and then go down, write a cross, and then it's a continuation of meiosis 1. And then next to meiosis 2, write an asterisk, and then go down, write the asterisk, and it's a continuation of meiosis 2.
I'm definitely confused. I'm just gonna do it.
I don't know.
Just rock with it.
I can't.
Okay, so here's meiosis 1. Just like we have prophase, prometaphase, metaphase, anaphase, and telophase or telophase in mitosis, we have all of these phases in both meiosis 1 and meiosis 2. You're like, well, that makes sense, Dr. Engel, because you told us meiosis 2 is identical to mitosis. We're just starting with half as much DNA. You're absolutely right. Okay? And now the things that happen during these stages happen during these stages as well, with some minor changes. So during prophase 1, what happens during prophase of mitosis? Yeah, Katie.
Chromosomes condense.
Chromosome condense. You better believe that happens here. Chromosomes. Wow, this room is echoey.
Ooh, the spindle forms.
Yes, the spindle. And this, we still call it the mitotic spindle. The spindle forms.
This is
Cor-
literally just mitosis.
Yeah, but
So
we
far.
need to add something. Oh yeah. Oh yeah.
Centrosomes go to the poles.
Yes. Oh, I'm out of space.
My notes are.
Just go under connect.
Centrosomes go to poles.
I did this terribly, but it's fine.
But some unique thing happens during prophase 1 that does not happen during prophase of mitosis. And that is homologous chromosomes. What we call crossover. Okay? And you're like, that's awesome. What does that mean? Okay? So still this similar things happen during prophase 1. The chromosomes condense like they did in prophase of mitosis. the spindle forms like it did during prophase of mitosis. The centrosomes migrate to opposite poles like they do during prophase of mitosis, but something unique happens. Homologous chromosomes, sorry, that should be plural, chromosomes cross over. Cross over. And this is where I would really like a different colored marker.
I would too.
Yeah.
We should go down there and
So crossing over.
oh, really?
I mean, we can always just take our markers back, and they are our markers.
They are our markers. So crossing over, right? You see that during prophase 1, we need to show what's actually going on here. Okay? True or false? A typical cell in your body has 2 copies of chromosome 1.
True.
True. Okay? And so we're going to draw this. Our 2 copies of chromosome 1. Okay? So you've got one that came from your dad and one that came from your mom. Okay?
Hey, why are
Mom.
those the symbols?
Well, I don't know. I don't know the origins of the symbols. But so you've got 1 copy of chromosome 1 that came from dad and 1 copy of chromosome 1 that came from mom. What happens during prophase 1 is that a portion of your paternal chromosome exchanges places with a portion of the maternal chromosome.
What? That's weird.
It is weird.
Like,
And that's what we call crossing over.
Wait, can you repeat that again?
Yeah, so a portion of the paternal chromosome switches places with a portion of the maternal chromosome. It is.
Is it the same portion? Like, you know how they
Yeah, ideally. Sometimes it's what we call an uneven crossing over. And so let's say more of the maternal goes over here than of the paternal that goes back. But ideally, it's the same portion and the same amount. Okay, so you just basically get an exchange of information. And you're like, well, how does that happen? But you have to remember, these are both chromosome 1, right? So your paternal chromosome 1 should be very similar to your maternal chromosome 1, right? I mean, there should obviously be some differences because your mom and your dad are not identical twins. But there are going to be a lot of similarities. So they can associate pretty well. Michelle.
Is it just a portion from
It can happen anywhere on the chromosome. And it happens on average 3 times per chromosome pair.
Weird. Like just
On average, so approximately 3 times per chromosome pair. That you'll get an exchange of information. Okay? And you're like, that's so cool. And why is that cool?
because it's weird.
It is weird. And what happens during meiosis? Yeah, Isa.
Well, they basically end up like switching, right?
They do. So during meiosis, you make chromosomes that have never existed before.
That's some weird stuff.
You make chromosomes that have never existed before. Because you got a set of chromosomes from your dad, which the chromosomes you got from your dad are actually a mix of your paternal and your paternal grandmother and your paternal grandfather. So your dad gave you chromosomes that never existed before your dad. Or maybe in your dad's siblings, like they're making the similar chromosomes. But your dad gave you, so your dad's chromosome 1 is a combination of your paternal grandfather's chromosome 1 and your paternal grandmother's chromosome 1, because this happened when your dad was making gametes. Okay? That his father's chromosome mixed with his mother's chromosome, and he created chromosomes that never existed before. Is that cool? And then when you make gametes, you're going to generate chromosomes that never existed before because they're part of your dad's chromosome and part of your mom's chromosome. It's so cool. That's so cool. All right, so that's what happens during prophase 1. Everything that happens during prophase of mitosis plus this crossing over, this mixture of genetic material from the maternal and the paternal chromosome. During prometaphase, this is basically just like prometaphase of mitosis. The kinetochore microtubules, what do? Well, the kinetochore microtubules grab the chromosomes. The the nuclear envelope dissolves. Right, these major events that happen during pro-metaphase of mitosis still happen during pro-metaphase 1. Okay, the nuclear envelope dissolves, the kinetochore microtubules make contact with the chromosomes so that they can then separate them during anaphase. Right?
Right.
We good?
Yep.
Okay. During metaphase 1, what happens during metaphase of mitosis? The chromosomes line up in the middle of the cell. During metaphase 1, they line up in a unique way. The homologous chromosomes, which now this is a term you've seen a couple of times. What does it mean? What are homologous chromosomes?
They're the same.
Yeah, they're the same. Both copies of chromosome 1. So they're not identical because one came from your dad and one came from your mom, but they're both chromosome 1. Okay? So the homologous chromosomes line up side by side. This does not happen in mitosis. In mitosis, you end up with a single file line of all 46 chromosomes. Okay? In meiosis 1, in metaphase 1, the chromosomes line up side by side. Okay? You create two lines that are side by side because the homologous chromosomes are lined up side by side. But it's not like all of your paternal chromosomes are on the left and all the maternal are on the right. It's random where they end up positionally. Okay? And so I'll draw this for you. The homologous chromosomes line up side by side. So here's our cell in metaphase 1. And here are our two copies of chromosome 1. Here are our two copies of chromosome 2. Here are our two copies of chromosome 3. And look at that. They are lined up side by side. Do you see that? And it's so cool. Like they're literally holding hands. Because at the site at which they crossed over, they're still connected together during metaphase 1.
Love it.
So they're literally holding hands. But this happens on average 3 times per chromosome, so they're holding hands, feet, and their heads are attached together. Right? Leaning in. Yeah.
How cute.
It's adorable.
Valentine's Day. We love it.
I know, it's beautiful. Okay?
They gotta save room for Jesus.
This is pretty cool, right? Okay, so during anaphase 1, the homologous chromosomes separate. So during the anaphase 1, the homologous chromosomes are pulled to opposite poles instead of the chromatids. So in anaphase of mitosis, the sister chromatids are pulled apart. In anaphase 1, the two homologous chromosomes are pulled apart. So that the maternal chromosome with some pieces of paternal end up on one pole, and the paternal chromosome with some pieces of the maternal end up in the other pole. Okay? And then what happens during telophase or telophase? Somebody remind me.
Envelope.
Envelope reforms.
Spindle dissolves.
Spindle dissolves. Chromosomes decondense. Spindle reforms. Envelope reforms. Spindle dissolves. And chromosomes decondense. And you're like, wow, this is so cool. Decondense. And then you know that there's another event that overlaps anaphase and telophase, and what is it? Cytokinesis, the cell splitting in two. So after anaphase 1, we now have 2. Okay, so the product of this, I'm going to write that, product equals 2 genetically distinct cells. Genetically distinct cells. Okay? All right? So that's the product of meiosis 1. 2 genetically distinct cells
As opposed to
With
identical cells.
half the DNA. Okay? Sorry, I needed to add the rest of that. With half the DNA. Half the DNA of what? The cell at the beginning. The germ cell. And the typical cell from your body. Okay? So 2 genetically distinct cells with half the DNA. And so, yeah.
It's about the crossing over.
Uh-huh.
So the cells would be different even if they didn't cross over, right? It just makes
Yes.
them more distinct since it crosses
Yep, absolutely. They would be different, and we're going to talk about that in a little bit as well. But yes, they would be different even if crossing over didn't happen. Absolutely. Okay? Cool? Cool.
Hold on, can you keep the bottom? Oh, actually.
Yeah. So I'm not even going to write meiosis 2 for you because I already told you meiosis 2 is identical to what? Mitosis, except you're dealing with half the amount of DNA to begin with. Other than that, it's identical. Okay? So I'm not even going to do that. We're going to go into what happens in between the two, and then how do we increase overall diversity. Okay? Any questions on this? Okay. So we're going to take a little bit of a break. Feel free. Yay! Shh. So we are now live again from Mr. Freeman's classroom. All right. So what happens in between meiosis 1 and meiosis 2? And the answer to that question is this. A very short, truncated interface. So distracting. So we have a very short, truncated interface that separates meiosis 1 and meiosis 2. Now we know that the bulk of the cell cycle of mitosis is interphase, right? We did this activity on Tuesday where we counted the number of cells, and about 50% of the cells that are actively mitotic are in interphase, right? So that's the bulk of the cell cycle, even in an actively mitotic cell. But we know the typical cell in your body is not actively mitotic. It's not growing and developing. It's left the cell cycle, right?
Right.
Okay. So in between meiosis 1 and meiosis 2, we have a short, truncated interphase. And in this interphase, there's no DNA duplication. And why does that make sense?
Because it has to keep it,
Yeah, meiosis 1 accomplished having the DNA. It already accomplished what needs to happen during meiosis to make gametes. You don't want to reverse that by duplicating the DNA, right? That's bad. So we have no DNA duplication during this short, truncated interface. We do have some cellular growth, which typically happens during G1 and G2, some cellular growth. Now this is shorter, so there's not as much opportunity for growth, but there is some. And it seems to be that we do duplicate the centrosomes, or the centrosome. Right, because after meiosis 1, each cell only has one centrosome, right? The centrosomes migrate to opposite poles in prophase 1, just like in prophase of mitosis. Do you remember that? We wrote that. I think it was either Carrie or Katie that reminded us of that. Was it Katie? That the centrosomes migrate to opposite poles. It wasn't you? Well, it came from behind. It came from behind me as I was writing on the board. I think it was from the middle. Anyways. So after meiosis 1, each cell, each of those 2 genetically distinct cells with half the DNA only have one centrosome. Okay, so it seems that we duplicate the centrosome during this truncated interface. We don't duplicate the DNA, but we do, it looks like, duplicate the centrosome. Trinity.
It is probably stupid.
Yeah, this well we talked about it on Tuesday. This is where the mitotic spindle grows out from. But I'll put that here as a reminder. The mitotic spindle growth point. It is referred to, this is more information than what you need to know for this class, but if you know more than what you need for the AP exam, you're in a good position. The centrosome is what's called a microtubule organization center, because it organizes microtubules. Even outside of the cell cycle, when you're just using microtubules to do normal microtubule jobs, a lot of them grow out of the centrosome. Okay, so this is what happens in between meiosis 1 and meiosis 2. We have a short truncated interphase with no DNA duplication, some cellular growth, and it seems to be a duplication of the centrosome. Okay? And then I told you we're not going to go through meiosis 2 again as we go through meiosis 2.0 because you know it's identical to what process? Mitosis. And we went through mitosis in detail on Tuesday. Meiosis 2, again, it is identical to mitosis with one exception. And what is that exception? It only has half the DNA.
Yeah.
So instead of having 46 chromosomes, you only have how many? What's half of 46? 23. Excellent.
Wait. Isn't it in pairs though?
Yes.
So they paired 23.
Yes, because the XY is a thing, right?
Uh-huh.
Wait. Okay, that makes sense.
Okay. All right. Any questions on this?
No, and that's the one that makes the one that's alone. Isn't that what determines the...
There is none that's alone. It's all 23 pairs. Even the X and the Y pair together.
So instead of 46 pairs, there's 23 pairs.
Well, there's never 46 pairs. There's 23 pairs, 46 total.
And when you have
And then when you half it, it's just 23 solo chromosomes.
Oh, so
No longer
Those
23 pairs.
ones don't
I know.
during mitosis.
I know, and on Valentine's Day, after anaphase, in anaphase 1, they separate them. Where they're holding hands and holding feet and have their heads touching together, they're ripped apart, never to see each other again.
That's pretty much.
You have to wait until Michelle comes back. Sorry. That I know is a rule because I've been emailed like 5 or 6 times to remind me. Please do not allow 2 students to leave the classroom at the same time.
It's usually one girl, one boy.
Yeah, 1 boy and 1 girl can be gone at a time, but not 2 boys and not 2 girls.
Senora. Senora.
I think they want to avoid like the whole situation of like, hey, do you want to go freshen up with me? You know? And then you go and you spend like 20 minutes like talking, and you've got somewhere where you can go and hide and nobody's really going to go looking for you, you know? So.
I used to
Yes,
do
because there's so many places to hide.
I know,
in school
the
the size of
school
a
is
peanut.
School the size of a peanut.
Not that small.
Have you seen Zoolander?
A large yes.
What is this, an institution for ants? How are we expected to teach the kids how to read if they can't even fit in the building? It's got to be at least 3 times this big. It's like a little model of the Derek Zoolander Institute for kids who can't read good and want to learn how to do other stuff good too. Great name for a building. This is in no way an endorsement of that film or a suggestion that you should watch it. I'm just... It is called Zoolander. Oh, Ben Stiller, yeah, and what's the dude? Owen Wilson.
I haven't seen it, but I want to.
He's Hansel.
You don't watch it.
So hot right now. Okay, so the next question we need to answer is this one. Why reproduce sexually? Why reproduce sexually? Why not just make clones of yourself? If you're rocking life, you're like, I got this thing figured out. If you make a genetic clone of yourself, they're gonna have that thing figured out.
And that is how the world.
And we can't have that.
A whole world of the long ones.
Okay.
Everyone needs more.
And we have basically 3 big answers for why. 1,
To have kids and
environments change. because environments change, there is a benefit to making offspring that are different than you are. They might be able to better face a changing environment than you can. Okay? So that's one big reason. Another big reason is to lessen the probability.
Because if two are making one.
Probability
Two are better.
of genetic illness.
Wait, how does that lessen the probability?
Because
Because we have the same genes and
yeah, you're building new combinations, right? And we haven't talked fully about Mendelian inheritance, but we talked about allele frequencies and the dominant and the recessive. A lot of genetic illnesses are inherited when you have 2 copies of the recessive allele. Okay? And so if you're mixing your genetic material with another individual, it makes it less likely you're going to get 2 of the same version of that gene, right? So it makes it less likely you're going to get 2 versions of the recessive allele. We see this in purebred dogs, right? Purebred dogs, genetic illnesses are way more common than they are in mutts. Okay, and again, it's because of a lack of genetic variability. So one big reason is environments change, and so there's some benefit to creating individuals that are unique, that have genetic combinations that have never existed before, and then to lessen the probability of genetic illness. And then the other one is to maximize the restart, and I'm going to put this in parentheses, maximize the restart that comes from producing, please stop with the whispers, producing new offspring. Okay, so even if you only reproduce what we call asexually, you still get somewhat of a restart when you make an embryo, right? It's starting its life anew. Now it's genetically identical to you, but anything that you've built up during your life may not be present in that individual, right? So anything that you've built up, like let's say you've got some kind of trauma, like physical trauma, that has reduced your vitality, right? That restart eliminates that trauma, right? Like let's say you shatter your femur, doing something epic like going skydiving,
More like trying to.
and you land improperly with your legs locked, and so it puts all your weight onto there, and it just blows your femur apart.
Oh
Okay?
my gosh, I am
Sounds fun.
Okay, and so
Your bone is launching your hamstring.
some breaks, breaks are going to remodel, right? Bone's going to remodel, but some breaks are so catastrophic that it handicaps you for the rest of your life. Well, forming offspring creates a restart. Even if you're forming an individual that was genetically identical to you, it's still going to get a restart. Anything that happened that was like a developmental abnormality and wasn't a genetic issue, that's all going to be erased when you restart, right? But in sexual reproduction, it maximizes this restart. Because not only are you restarting with a new individual that may not have the developmental or the issues that you've developed during your lifetime, but you're also restarting with an individual that's different than you are genetically. So which means you can restart any developmental or life history issues, and you can restart any genetic issues.
Excellent.
Okay, so it maximizes the restart. So that's why there's a benefit to sexual reproduction. Now it's not faster, right? It'd be way faster to just reproduce asexually throughout your entire life, right? Just keep budding off new individuals that are genetically identical to you. Yeah.
Wait, can you explain the restart again?
Yeah, so the restart is basically erasing everything that's accumulated during your life, right? So let's say you're at the end of your reproductive years, which for humans, it's probably somewhere around 50 to 60, depending on the individual. Some people can push past 60. Now males, Clint Eastwood had a child in his 70s, right? So that's not unheard of. But for females, you're
serious?
yes, you're probably looking at like somewhere between 55 and 60 is about the end. But let's assume you're like in your late 40s, right? And so you're about at the end of your reproductive years.
Yeah.
Having an offspring gives you a reset, right? It allows you to produce something that has half of your genetic markers,
Yeah.
but none of your, developmental issues that may have happened because maybe your parent didn't consume enough nutrients while you were in utero, right? Or you didn't eat enough as a kid, right? It gives you a reset of that. And then on top of that, it's genetically distinct from you. So they're gonna, just by the feature of them being genetically distinct, they're gonna develop a little bit differently than you did. Because they've got a different code, a different program.
Good stuff.
Pretty good stuff. Just
Beautiful.
All right, any questions about this? Because there's only one more thing we need to talk about. It'll probably take us about 7 minutes, and then we should have about 20 minutes to work on our corporate time.
Wait, what are we gonna do? Okay.
we have whole 20 minutes.
Okay. All right.
I really
Love it. I love it.
So I think the first line, and I know like I'm erasing now, so I can't really be doing anything else, should be something about, let's talk about fiddler crabs. Bum, 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 and then we can get in and talk about
Yeah, it's like.
fiddler crabs. No, you do fiddler crabs instead of the bum, bum, bum that time, and then the rest of it becomes the bum, bum, bum. Bum, bum, bum. But the first time you do it, it's fiddler crabs, And then it goes to the bum, bum, bum. Yeah. Yeah. Oh, yeah. Oh, yeah. Okay. The last question.
Oh yeah, oh yeah.
The last question is this one. How does meiosis increase diversity?
Oh, huge. If you
How does meiosis increase diversity? We're not talking about sexual reproduction right now, because you're like, oh, you take a gamete from one individual, fuse it with a gamete of another, right? That's obviously going to increase diversity. We're talking about just the process of meiosis. Just the process of meiosis. How does it increase diversity? Emma.
When the two chromosomes, the parent and the maternal one, switch the thing.
Yes!
crossing over.
So crossing over creates, welcome back, unique, or did somebody leave?
That was Tori coming in.
Okay. I just assumed that that's what it was, Tori returning, but it could have been somebody like, man, I'm out.
He
Who
turned
would that
around, let
be?
me just ditch. Has someone ever done that in their college class?
Oh, all the time.
Yeah.
Right, people, I tell them, I tell them to put their phones on the bench, and if they get a call that they need to take to leave. So it doesn't happen very often. And then some people are just like, you know what? I'm done with this today, and they just bounce.
That's hilarious.
So crossing over creates unique chromosomes, right? Chromosomes that never existed before. It takes part of your paternal chromosome and mixes it with your maternal. And so ideally you can get the best of both worlds. Or you could get the worst of both worlds, right?
You never know.
Like, let's say your dad is a carrier for some genetic illness, meaning he doesn't have it, but he's a carrier. One of his copies has whatever the version is that leads to that genetic illness. Carrier means you're not expressing it because you have one normal copy of the gene, but you have one abnormal copy of the gene. And so you're a carrier. You can pass it on to your offspring, but you're not actually expressing it, right? So you could be like, your dad could be a carrier for one genetic illness, and your mom could be a carrier for the other, and then you mix the two chromosomes, and then now you're a carrier for both. Yeah.
So, I know this family. Is cystic fibrosis that, like, the lungs thing?
Yes.
So they had, like, a 1 in 4 chance of having a kid and then, so their first kid had it, and they're like, oh, it won't happen again, and then both of their boys have it.
Yeah, I mean, that's the thing is like, you just assume if it's a 1 in, you think about it, well, if it's a 1 in 4 chance and our first kid has it, then there's less a chance for the second. No, there's still a 1 in 4 chance, right? It's like having a male child first, and you're like, well, it's 1 in 2, so it's going to be more likely we're going to have a female child next. No, it's still 1 in 2. It's like flipping a coin. The previous has no impact on the future. Okay, so that's one. The other one is called, random assortment of chromosomes.
Oh, about the kids, I know.
Chromosomes
I bet I knew.
during
I just wanted to make some comments.
metaphase 1. There's no way you can be thinking properly about what I'm writing while also having this conversation.
It was about this kind of stuff, actually.
Okay, so random assortment of chromosomes during metaphase 1. And here's what I mean by this. So let me draw you a cell. And I'm just going to do 3 chromosomes. So let's assume we have an organism that has 3 chromosome pairs instead of the 23 that we have. Okay? 3 pairs. 3 pairs.
Chromatids.
Thanks. So we've got to line up these 3 pairs of chromosomes during metaphase 1.
Oh no.
Okay, we've lined up these 3 pairs. Now, here's what can happen. You could have the paternal here on this side, and this one could also be the paternal, and this one could also be the paternal, right? Which would make the 3 maternal all lined up on this
Where are
side,
you going? Oh, I see. Yeah,
right?
yeah.
So that's an option of how they could have lined up. And then when they separate during anaphase, the ones that are on the right are going to go to the right. The ones that are on the left are going to go to the left, right? Does that make sense?
Yeah.
Okay? So it could be like this, where all of your paternal chromosomes are on the left, all of the maternal are on the right, but how else could it be?
Mixed.
This one,
One.
the maternal could be here and the paternal here, right? Or we could have had something like this.
Or 2, yeah. And that's
Now, if we put all the paternal on the right and all the maternal on the left, that's the same thing, right? It's going to accomplish.
so those are only options, yeah. 5th, 3.
Okay?
Okay.
Is that cool?
So cool.
Yeah.
So we have different ways in which these chromosomes can assort during metaphase 1. And that's going to create some randomness to our gametes and increase diversity. So, how many options are there at each position?
2.
2, right? The paternal could be on the left or the paternal could be on the right, right? We've got 2 options. So every site gives us 2 options for how the chromosomes are going to be lined up. For chromosome 1, we have 2 options, paternal on the left or paternal on the right. For chromosome 2, we have 2 options, paternal on the left, paternal on the right. So how many different combinations can we get in our gametes because of the number of pairs that we have?
6? I don't know.
We have 2 options at each site raised to the number of pairs. So 2 to the 23rd different combinations that we can get from random
Very
assortment.
thick number.
I don't even know what that is. Isa.
When they say like, oh, the dad like chooses the gender, you know, or like not chooses.
Well, that is true because the mother can only give an X chromosome, right? Female mammals they don't have a Y chromosome, so you can only give the X. And so gender is going to be determined by which chromosome that the male contributes to his gametes. Okay, so 2 to the 23rd power is 8,388,608. So we have 8,388,608 different gametes that you can form just based on random assortment. And you're like, that's so cool. But males are going to make more than that number of gametes in their life. Way more than that amount. Females aren't going to make anywhere close to that number of gametes, but males will. But then when we add crossing over as well, right? Because crossing over is going to create unique chromosomes. There's basically an unlimited number of gametes that you can make. Basically. Which means it is virtually impossible for you to make 2 gametes that are genetically the same. Okay, it's virtually impossible that you could ever in your entire lifetime produce 2 gametes, even if you produced 1 at the very beginning of your gamete production, which in males would be at puberty and females would be in utero. Females start producing gametes while they're still in the womb. Males don't start producing gametes until puberty. But let's say even one that you produce at the very beginning, one that you produce at the very end, it is virtually impossible that they could be genetically identical. Females also stop producing gametes in utero. By the time you're born, you have all the gametes that you will ever have in your life. But they're all in a state of arrested development. They're frozen in prophase 1 until cyclically, every 28 days, one of them progresses through and finishes meiosis. Okay, Tori.
Okay, is that why males can have children a lot older than females can because they start producing gametes later?
Yes, yep, that's part of it. And then, so females, and this is for all mammals, you've produced all of your gametes that you'll ever produce in utero. So by the time you get to 50, those cells are 50 years old, right? They're arrested in a particular point in development, but they are 50 years old. Whereas the gametes a male's producing at 50 years old, those are new cells. So, yep. Emma.
This is kind of off topic, but you know, like when women freeze their eggs, if they don't want to have children, what exactly are they doing?
They're taking these, they extract these gametes that are arrested in prophase 1. So they'll extract them and then they'll freeze them and to wait until you want to activate them. But then once you do that, you've got to activate them in what we call in vitro. You've got to do it in the lab and then you can implant the zygote afterwards. But once you remove the cell from the female, you can't put it back in until it's already fertilized, and it's going to send the signal to implant. So that's what you're doing if you, or even if you donate eggs, like you donate eggs to science, or you donate eggs for couples that can't, like the female just doesn't produce eggs at all, right? And that's the reason she can't get pregnant. She just doesn't produce eggs. So, all right, but that's what's going on there. But inside of your body, they're not frozen. They're still accumulating issues, right? They're frozen at a particular stage in meiosis, but they can accumulate issues. And that's why the gametes that females make later in life aren't as high quality as the ones that they make earlier in life. Okay. And certain genetic illnesses become a lot more common. All right, any other questions? Okay, so we've talked about meiosis, how it differs from mitosis, why meiosis exists, what it does, what its purpose, how it increases diversity. We've talked about gamete production, and that in female mammals, all of your gametes you're going to have for your life are already present in utero. In males, you don't start producing gametes till puberty. We talked about that in males, the gametes that you make are basically all the same size. You take one germ cell, you get 4 mature sperm eventually after they mature. In females, you take 1 germ cell and you get how many ova? 1. And then 3 polar bodies that can't really be used at all. All right?
Wait, when you're on your period?
Yep.